In this chapter, we're going to look at the specific transcription factors belonging to a few families. There are four major things in this category: zinc finger, leucine zipper, helix turn helix, helix loop helix. This could explain more than 80% of eukaryotic motifs. Several thousand different transcription factors contain zinc as an essential element of their DNA binding domain. The classic zinc finger motif was first found in 1985 by Aaron Klook in MRC in United Kingdom. This one is present in TF3A, which is involved in 5S rRNA transcription in Xenopus laevis. This protein has a 9 repeat of 30 amino acid, which has zinc. Basically, they are different sequences, but share 2 cysteine and 2 histidine. The residue numbers between 2 cysteine is 2 or 4, and between cysteine and histidine 12, and between 2 histidine is 3 to 5. Actual crystal structure was obtained from the X pin from the Xenopus laevis. This transcription factor is kind of large, having more than 37 zinc finger, 31st among which was determined for their structure. We are looking at the crystal structure. Basically, 1 to 10 make antiparallel hairpin motif. 12 to the 24 forms 310 helix, where hydrogen bond is made between every third, and also hydrophobic interactions formed by phenylalanine 10 and leucine 16 locating between beta and alpha. And interestingly, zinc also contributing to make hydrophobic core. Zinc doesn't contribute to direct DNA interaction, but involved in making over zinc finger structure. Actual zinc finger and the DNA complex came out of different zinc finger, which is GIF268 protein. We are looking at the 3 zinc fingers aligned for the 2 beta followed by alpha helix, and also the DNA that could be recognized by this zinc finger. 3 zinc fingers binds in tandem to the major group of DNA. The zinc fingers have the same structure and bind in a similar way with the end terminus of alpha helix pointing into the major group. Each finger makes its sequence-specific contact with 3 base pairs. The 3 tandemly oriented zinc fingers therefore binds to 9 consecutive base pairs in the major group. The first beta in hairpin does not make any contact with DNA, whereas the second beta strand contacts sugar phosphate backbone along one strand of DNA. This slide shows detailed view of binding of a second zinc finger in the previous picture to the DNA. The 12 residues between the second cysteine zinc ligand and the first histidine ligand of the classic zinc finger motif form the finger region. Structurally, second beta plus 2 residue between second beta and alpha helix and plus the half portion of alpha helix. There are two types of interactions. Sequence-specific interactions with the DNA is made by arginine 46 and 49, and non-specific interactions are formed by the other three residues. The description I made about the zinc finger binding to DNA has also been found in other classic zinc fingers. In this slide, try to explain how zinc finger domain could bind to the DNA in different DNA sequences. The residues in the N-terminus of alpha helix in zinc finger regions are numbered 1 through 6, and the residues immediately preceding the alpha helix is numbered -1, -2, something like that. Amino acid residues and nucleotides that makes sequence-specific contacts are colored in both sides. In spite of structural similarities between zinc fingers and their overall order of binding, there is no simple rule that governs which basis the fingers contact. Zinc fingers are randomly arranged in the major group, and successive zinc fingers bind to successive subsets of 3 to 5 base pairs along the DNA. Two hydrogen bond formed between arginine and guanine is frequently observed in sequence-specific interaction. 4 cysteine zinc finger exist in glucocorticoid receptor. The glucocorticoid receptor is a member of a family of transcription factors that include many hormone receptors shown here. All members of this family have a highly conserved DNA binding domain containing zinc that consists of about 70 residues and that binds to the activating element of DNA called hormone response element. DNA binding domain in red have highly homologous sequences among these receptors, while the ligand binding domain in blue color are more variable. Amino acid sequence of the zinc containing DNA binding domain of the glucocorticoid receptor. There are 2 zinc atoms within this domain. Here, each bounds to 4 cysteine residues. One of these stabilized the recognition helix in red color that provide sequence-specific binding to DNA, and the other, zinc, region contains a loop in purple color that is involved in formation of dimeric molecule. You can see in B, DNA region that binds to the glucocorticoid receptor called glucocorticoid response element. The two palindromic half sites are separated by spacer regions in the middle. This shows schematic diagram of DNA binding domain of glucocorticoid receptor. Two zinc binding regions are colored red and the pink, and connecting loop in blue color. Each has one alpha helix here and here. There is no beta strand in this structure. Two alpha helices are part of compact globular core, shown in gray color. The dimerization loop, shown in purple color. The zinc atoms and residues between the zinc ligand form protrusion from this globular core. Structure of the complex between dimeric glucocorticoid receptor molecule and DNA fragment are shown here. The recognition helices of dimer, brown and red color, are positioned in the major group. The distance between them, which corresponds to 1 turn of DNA helix, is fixed by the dimerization loop, purple one. This region undergoes a significant conformational change when the dimer binds to DNA. An alpha helix in the first zinc motif provides the specific protein-DNA interaction. So you can see here such sequence-specific interactions between DNA and recognition helix of the glucocorticoid receptor. Residue lysine 461 and valine 462, arginine 466 makes specific contact with the edges of the bases in the major group.